Today I'm going to show you how to key out a sedge. And sedges are really interesting because there's a lot of them. There's a lot of species in New England. And so that means that every time you go out, you can probably see a new species of sedge. And they are, once you start learning a few characteristics about them, they are pretty distinctive. And you can almost tell what species it is, just by looking at it without keying it out. Some species you have to, and there's varieties and whatever. The other thing is there is a theory that you can tell species just based on leaf morphology. So and I sort of believe that because as you get used to seeing plants more and more, you can tell these really like distinct differences, even if they're minor, between particular species, and that helps you when you're going out in the woods and you have to like walk through an area pretty quickly and figure things out. You can like knock down a few species that you already know, and then of course you have to verify the species, especially of a Carex, when they're in fruit. And so that's what we're going to do today. We're going to look at this dried specimen that I took back in June of 2024, dried it in a plant press, and now I have these parts and I'm going to verify the species, and we're going to be using two different keys for that. So thanks for tuning in to another episode of Only Plants. Today we're going to key out a sedge. And so this sedge I found in New Haven, Connecticut. I think I know what the ID on it is, what the specific species is on it, but I'm going to show you some pictures before we get into a key. We're going to use the key for New England because that's where we found it, and that is the Flora Nova Anglae by Haines, and so that was published in 2014. So we're going to use that key, and then we're going to use another key, the Stanley key, which is a key that was put out by Lisa Stanley, was put out by New England Botanical Society, and it's actually free. You can download a PDF through their website. I'll put their website in the description, and you can download it. I got it printed at Staples for like 30 bucks, but it is a really valuable resource. And so maybe we should let's start with that one. So here's the cover of it, and I'll show you the sedge first. And so these are my observations from iNaturalist, and they're descriptive in that I have a measurement tool in my finger, so I can tell about how big those were in life. There's also some pictures in here that show sort of like a more up close, and there's a few identifying characteristics you can just take from this. You can see the leaves are relatively flat. You can see how wide they are. You can also see what the perigynia look like in life. You can see some veins there on the perigynia. You can see the male part of the flower and the female part of the flower. And so these are good photos to have. I also have the actual, so there were enough plants here. I don't usually do this, but I happen to take a sample. And so what we're going to do today is we're going to use that sample to actually key out this species again and just verify that it is the species that I think it is. Normally I don't take samples from the woods, but I liked this plant, and I just liked the way that it looked. So I have a dried specimen here, and it's been in cardboard in a plant press, and it's been probably drying since mid-June. So we do have some really cool things here. And today I'm going to be showing you up close photos with the Beaverlabs Darwin 2 microscope. And so that actually connects to my iPhone, and hopefully I'll be able to show you some really up close ID and parts of these flowers, so that hopefully this will help you in the future identify Carex in the woods. So the first part of the key asks, is it a single spike or are there more than one spike? And so if you look at this plant, you can see that there's multiple spikes here. So it's not just one. Sometimes you will get a plant that just has one spike on it. So it usually be like a terminal fruiting cluster. So we're going to go with spikes more than one. Several small spikes, all alike in sessile, achenes lenticular, perigynia flat or plano-convex. So lenticular means like a lens, so that would be flat. So, or the other option is spikes cylindric or globose, generally peduncle, generally with male and female flowers on separate spikes or all spikes appearing female. The terminal spike with some empty scales at base, achenes lenticular or triogonous, perigynia biconvex, triogonous, or round. So let's look at this perigynia up close and see, the spikes are cylindrical, so they're pretty evenly spaced along the length of the fruiting body, or globose, so that would mean like a roundish, more globe-like structure. Generally peduncled, so that just means that there's a little stalk here, and so what they're talking about is this little stalk here coming off the stem. This is a peduncle with the fruiting body on top of it. So you usually have, sometimes the length of those can matter. Like these ones are, the ones at the top are relatively shorter, but if you see a peduncle, then it counts. If you don't see one, if they're all sort of like sessile right against the stem, that would be a different ID characteristic. So here they are up close, and you can see that they are sort of like, there is maybe that triogonous nature to them. So let's go with that for now. So in that key, it says the last part of it is achenes lenticular or triogonous. So these achenes aren't lenticular, but you can see that they have sort of three sides to them, so like a triangle. So they are triogonous achenes. If you look at these leaves are approximately 5 to 10 millimeters wide, so they're underneath the amount here. So we're gonna go with the part of the key that says leaves less than 2 centimeters wide. So we went leaves less than 2 centimeters wide. That brings us down into number 9, male and female flowers on separate spikes. The terminal spikes all are mostly male. So if you look at this, these inflated fruits are the female parts. The male part is up here. Sometimes they'll be intermediary in between this female part. Sometimes they'll be on a separate stalk altogether. These are occurring on the same stalk, but they're separate in terms that the male part is on its own peduncle stem versus the female part. And so when we look at this, this says male, the choices are male or female flowers on separate spikes. The terminal spikes all are mostly male. Or the other one is all spikes predominantly female, cylindrical, male flowers present only at the base of the terminal spike. So this is a good example because keys can sometimes be very misleading in that, in number 9 is a good example of this. So all spikes predominantly female, which this is, mostly female flowers, cylindrical, which they are, male flowers present only at the base of the terminal spike, which this is, right? But then when you look at, if you look at the species that are present in this, they don't look like this plant at all. But this one also works. Male and female flowers on separate spikes. The terminal spikes all are mostly male, so they are. So all of these terminal spikes are male, and they are on separate spikes. So I'm going to go with number 9, what would be like 9B here, because I've looked at these species in here and they don't look to be very accurate for what this plant looks like. So flowering stems elongate, not hidden. So these are, you can see them obviously. So we're going to go with that one. The other option there would have been very short, hidden at the base of the plant. So these are obviously above the plant based on the photos we saw before. Subulate just means like long and narrow. So the sometimes like folliculata, Carex folliculata would have really long perigynia. So these are not subulate. That brings us down to 13. So all spikes drooping. We can see from the photo that the spikes were not drooping. Spikes were erect, at most the lowest drooping, which maybe with that long peduncle on the lowest perigynia spike there, that was possibly. But so we're going to go with 13 here. Perigynia biconvex, which we know it's not, so that means like two-sided. Um, and we know that it's triogonous. So Perigynia round or triogonous, rarely triangular in cross section. And that brings us to 16. So Perigynia hairy or scabrous. So that means that you'll either see hairs, or they'll be kind of like spiky with shorter, stiffer hairs on them that are you can kind of feel them. Or the Perigynia is glabrous, which just means it's smooth. So let's look at the Perigynia up close and just see if it has hairs on it or not. So based on this enlarged example, you don't see hairs here. Some other species you'll see like it's almost like fuzz on the Perigynia, but you don't see any of that here. So we're going to go with glabrous for this one. And so if we do that, that brings us down to 18, Perigynia beakless or short-beaked, or Perigynia distinctly beaked, 1 millimeter or more, bidentate nerved. So bidentate just means like forked at the top, so you'll see like a snake's tongue. Uh, Perigynia beakless or short-beaked, less than 1 millimeter. So let's look at the beak of the Perigynia. So here's the beak is just like the top of the Perigynia here, and you can see that it's not forked. And I would say that it does have a beak, and it's probably, I'm not going to measure it, but it looks very short. It's almost like a nub on top of the Perigynia itself. So I would say that this probably is considered not beaked. Let's see another one. Let's see. So, and if it was, it's less than a millimeter for sure. And so when we choose that short beak, generally less than 1 millimeter, we're going to go to group 18 on page 132. And so what that did is it takes us to the group for this species. Because this genus is so big, they break it into like a bunch of different groups. And so now we're going to start looking at the really fine details on what species this could potentially be. So you can see from the photo that leaves in volute, that just means that they're curled up on the edges. So kind of like a U. And the leaves on this were flat. So we're going to say they're not involute. 2 millimeters wide or wider, which we agree with. They're like 5 or more. So we're going to choose that lead there. And then it goes down to number 3, leaves, sheaths, and bracts with short hairs are hispid. So we can look at the leaves on the dried sample and see if there's any hairs on them. So when you look at a leaf sheath, it's going to be where the stem, the flowering stem comes off the main stem, and there'll be a little sheath there of like, almost looks like vellum or something. And the way that that's grown on that plant is very distinctive for the species usually. So this one doesn't have any hairs on it at all. I can't see anything there that would be considered hairy or hispid. So it looks pretty smooth. So sheaths and leaves smooth. And then it brings us down to this next lead, which is about the Perigynia, triangular in cross section with flat nerved sides, or Perigynia triagonous or round, which based on the photos that we've been looking at, they all appear to be round. Now it brings you to 7, stems much shorter than the leaves, Perigynia ovoid, or stems as long or longer than the leaves. And so these stems were longer than the leaves. So we're going to go with that lead. That's going to take us down to scales dark purple or brown or red, Perigynia two-nerved, or scales pale or merely red flecked. So let's look at the scales. So in this enlarged video of the Perigynia, you can see the Perigynia here in green, and then the scale is this little thing that comes up sort of in front of it along the stem, and so you can see that this one is white on both sides and it has a green strip down the middle. And how this scale is formed here, if it's like shorter than the Perigynia, longer than the Perigynia, sometimes they'll be almost absent, sometimes they'll be like dark brown, sometimes they'll be rounded. And so this little tip here is important to look at as well. You can see another one here, and there's some more up here. So you can see these scales sort of like wrapping around the Perigynia. This is another part of the key where, scales dark purple or brown or red, which it's not. And it has, you couldn't really see, there's nerves on there, but there's definitely more than just 2. scales pale or merely red flecked. So I didn't see a lot of red. I guess there's some red along the edge of those. They're mostly pale. They have that green strip down the middle. But when we look at photos later to confirm ID, we'll look at that scale and make sure that they look like the photos that we're looking at for other species. So lower sheaths brown, perigynia rounded or tapering. So that is accurate. And then we can look at the sheaths here. You can see these sheaths on this plant, they are all brown. Um, and then perigynia many, which we do have a lot of perigynia within each spike, typically more than 40 on the lowest spike, inflated, ovoid, densely packed. So let's just see, more than 40 on the lowest spike. So the lowest spike is just going to mean the lowest spike that forms along the flowering stem. But let's take this one, and we're going to take it apart, and we'll see how many are on here. So there's about 37 here, which I mean, I feel like that's within the margin of error for something like this. Maybe it wasn't the exact lowest one, but we're going to go with this Carex granularis. All right, so we landed on Carex granularis for that. I hope that was helpful in terms of like looking at some stuff. But so let's do it through the Haines key now and see how that one varies. Sometimes keys can differ based on who makes them and sort of uh, how they work. I think we should do it. Let's do it from the book. Hold on one second. All right, so here's the hand key. Similar setup. It's going to take you to a group and then be more specific from there. So spikes entirely staminate. So that means that there would be no perigynia present at all. So those female inflated parts wouldn't be there. You just have male stems. And they're usually yellow and have pollen on them and stuff. So we're going to go with 1B. At least some of the spikes partially or entirely carpellate, which is true. So carpellate is another term for female. So we're looking at those perigynia to be present. We have them. Spikes one per stem. Rachilla often present in perigynium. So we have one, we don't have one spike per stem. We have more than one. So we're going to say spikes normally 2 or more per stem, except depauperate individuals. Rachilla absent from perigynium. So rachilla is like a zigzag stem inside it. Stigmas 2 or stigmas 3. So we'll have to look at, with this key, it's kind of hard because they're asking you to tell how many stigmas are there. Sometimes the stigmas fall off, they're deciduous, so they go away. So you can't tell. We're going to let's look at this and try to see if we can tell. Hold on one second. All right, so you can see stigmas here, and it looks like there's 2. Um, but some of these other ones, like the stigmas, have fallen off of this, and so you'll see either 2 of these, the really the female pistils coming out of the perigynia, or you'll have 3 of them. So usually the way that I identify this when I use that particular characteristic is if I find one with 3, I assume they all have 3, and usually you'll find like more than one with just 3. But I sort of take what the if they all have 2, they have 2. So it's kind of like if I find one with 3, I look closer to make sure that I'm going the right way in the key. And even if I make a mistake, I might go back and look through the other part of the key. So that's one hard thing to be able to identify with a Carex. So it looks like there's 2 stigmas, but the achenes are not lenticular or biconvex. So we're going to go with this triogonous lead here because we know that those achenes are triogonous and not these other 2 options, even though we can't really tell how many stigmas are there. So the next one is Perigynia pubescent or scabrous with hairs, scabrils several times longer than wide. Perigynia glabrous. We know from those other ones that we do have glabrous perigynia. Bract of lowest carpellate spike consisting of a blade only, the sheath absent or extremely short, usually not exceeding 4 millimeters, or the bract absent altogether, or the bract of lowest carpellate spike consisting of a blade and prolonged closed sheath, usually longer than 4 millimeters. So Back to the lowest carpellate spike consisting of a blade only. So the sheath, we need to look at the sheath on the leaf blade. All right, so the next part of the key is saying bract of lowest carpellate spike consisting of a blade and prolonged closed sheath, usually longer than 4 millimeters. So you need to look at your lowest female spike here, this fruiting part, and you need to look at the leaf sheath that's coming off it. Usually this is called the bract. So this is the bract. It can be varied sizes. But when you pull it down, you want to see if there's a sheath on it and how long that is. So this is saying longer than 4 millimeters. And so there is a sheath there. I would guess, yeah, it's probably longer than 4 millimeters. There you go. You can see that little thing sticking up there. So there's a sheath holding this little perigenia down on the bottom. And so if we choose that lead, it takes us into group 7, and it asks about the style. So the style is what we were looking at before, and basically the stalk that connects the stigma to the ovary. And so we were seeing some that were persistent on there, and then we saw some that seemed to be deciduous. Now that could be in part because I dried it and they fell off. So that characteristic doesn't really get me anywhere that's valuable to me. But if I pull out a measuring tool, we got our measuring caliper here, and we're going to take one of these. Basically says the break here is perigenia 3.4 to 19 millimeters long, or 1.5 to 6.8. And so these are probably going to be the second one just because they're so tiny. But if we measure, if we look at these, we can see that they're probably 2 millimeters or less. So it's going to fit in with our 1.5 to 6.8 millimeters long. So we're going to choose this lead. So this is the style deciduous from Rakeem, perigenia 1.5 to 6.8. Now we're going to get into a different one, and this is going to say leaf blades V-shaped or U-shaped in cross-section, at least when young. Adaxial surface of blades without two marginal veins that are prominent, more prominent than the midvein. So adaxial is the top, right? That's a good way to remember it. Abaxial is bottom. And so the surface of the leaf blades without two marginal veins. So we'll look at our photo again. Here's the leaf, the actual dried leaf. There's not two major marginal veins there. What normally you'll see is two. I guess I do see two there. So what they're asking about here is there's usually two marginal veins that'll be on either side of this that are much stronger than the other veins. And so this one kind of looks uniform across it. And from the photo, I would imagine that before this got flattened, it would have been more of a V-shaped cross section of the leaf than a U-shape. So if we go this way, and we get down into bracts of inflorescence consisting of only a sheath, perigenia green, maturing, glossy, dark brown, or at least the lowest bract of the inflorescence with a short blade, perigenia variously colored but not glossy dark brown at maturity. So, and we know that the perigenia were variously colored, but they're not glossy dark brown. They're very green at maturity, is what we've been seeing. That brings us into this next one, lower perigenia in each spike, horizontally spreading to reflex, the carpellate spikes, globose to short ellipsoid or short cylindric, or lower perigenia in each spike, ascending to spreading ascending, carpellate spike, short cylindric, or ovoid to narrow cylindric. So if we go back to our actual dried specimen, they're like erect, standing up straight. I wouldn't call them horizontally spreading, so that would be more of a right angle coming out. These are more like ascending. And so that would put us into this ascending to spreading ascending. So 8B, 9A, abaxial leaf blade of the surface of the perigenium, minutely papillose plants colonial from long rhizomes with strongly glabrous leaves, or abaxial leaf blade and surface of Perigynia without papillae. Plants loosely to densely cespitosum short rhizomes, colonial from long rhizomes, and cryi with green to moderate glaucous leaves. So this means that the bottom part of the leaf blade and surface of Perigynia is without little bumps. So papillae is little bumps. And so we've looked closely at these. We still have one up on the screen. You can see there aren't little, you'll see little raised bumps on the surface of that Perigynia. That brings us to 10. Perigynia rounded at the base, apically at the top with convexly rounded sides, often with red, brown, or yellow streaks and spots, or Perigynia tapering to the base, typically with flat or somewhat convex sides without streaks or spots. So we're looking at a rounded, these are rounded on the base, and they also have little red flecks there. So we're going to go with um, granulares and A, and that puts us into this sort of like subsection. Staminate spike on peduncle 1 to 35 millimeters long, shorter than to barely exceeding the uppermost carpellate spike. Stem cespitose on short inconspicuous rhizomes. Lowermost spike usually arising from the upper half of a reproductive stem. Or staminate spike on a peduncle 9 to 76 millimeters long, overtopping the uppermost carpellate spike. Stems produced singly or a few together from elongate rhizomes. Lowermost spike usually arising from the lower half of the reproductive stem. So we have to measure the staminate spike, which is the male part, and the peduncle on it. So let's do that. So there's that male staminate spike there, and the peduncle looks like it's, I don't know, 1, 2, 3, 4, 5, 6, 7, 8 millimeters maybe. Here's what they mean by that. You can see the staminate spike here just barely makes it over the carpellate spike, the female part. So there's only 2 species left in this key, and one of them is cryi, which well overtops the Perigynia. But when you look at this one, for example, there's some variation here. So this one is this one's almost twice as big as the Perigynia on the same species. So it's not always accurate. So you have to go look at photos and see from this point if they're accurate or not. So here's what we think it is, granularis, and it looks very similar to what we have. If we go back and look at cryi, this one, not only does the achene look a little different, when we were looking at those, the scales, so look at the scale on this. It has a point and then it's almost rounded on each side here. And that's not, and they're a little bit more brown than what we saw before on the other one. There's granularis, the limestone meadow sedge. And let's see if we have a photo of the scales here. So a little bit more similar to what we were looking at. Let's go to iNaturalist. So interestingly, if you click on this particular species, it actually brings up a bunch of really good diagnostic photos of plants you'd want to be looking at. And from here, you can look at, find anything you need about this species. So here's a good example of what I was talking about with that. It's well extended beyond the actual carpellate part of the species. And all right, here's some scales. So there you go. There's our scale there with that green line that runs down the middle, and it's more like white and pale on the outside. But I think this is it. I think we got Carex granularis limestone meadow sedge. So I hope this was helpful for you to learn how to start identifying Carex species. If you're interested in the Beaverlabs, Darwin 2 Microscope. There's a link in the description for this. Uh, I'm a paid partner with that one, so you can help me out by buying one of those. They're pretty cheap, $8. If you have any questions or you want me to look at any sort of Carex species you have and give you my opinion on what species it might be, um, drop the iNaturalist observation in the comments. I'll go check it out. Uh, like, subscribe, follow. Appreciate you. Thank you so much. We'll do this again.